Dear students, today our topic is about population interactions. The main objectives of today's lecture are to make students aware about the various types of population interactions, to make you understand how population interactions can be positive, negative as well as neutral. First of all, we will start with what we mean by population interaction. Population interaction refers to the effects that organisms in a community have on each other. Since all living organisms in an ecosystem are interdependent, the associations existing between them influences their survival and the functioning of ecosystem as a whole. Interaction can either be within a single species known as intraspecific interaction or between different species known as interspecific interaction. Population interactions are mainly based on three factors. Number 1, nature of food and mode of obtaining it. Number 2, kind of space needed. And number 3, habits. On the basis of nature of interactions, there are 5 types of population interactions. Number 1, interspecific competition, negative. Number 2, parasitism, predation, herbivory, negative-positive. Number 3, commensalism, positive-neutral. Number 4, mutualism, positive-positive. Number 5, amensalism, negative-neutral. Positive, negative or neutral symbols represent the outcome of interaction between the interacting species. Let's start with interspecific competition. Interspecific competition, represented as negative is interaction which is considered to be reciprocally negative interaction for both the species involved. Mill 1961 and Odum 1959 defined interspecific competition on the basis of cause of competition known as exploitative competition and effect of competition known as interference competition respectively. According to Mill, competition is endurance of 2 or more organisms to gain the same particular thing or to gain the major part of that. Each wants from the supply of a thing when that supply is not sufficient for both or all. While Odum defined competition as 2 species are in competition if population of each species adversely affects the growth of other in the struggle for common resource. Lotka 1925 and Volterra 1926 independently made first attempt to model interspecific competition. Lotka-Volterra assumed that the growth rate of each species would be decreased as the population of its competitor increases. Thus, the impact of one species on the growth rate of other species is expressed as the modification of logistic equation. Each equation includes a new term, the competition coefficient, alpha 1 2, which describes the effect of second species on first, and alpha 2 1, effect of first on second. Modified logistic equation are represented as this. dn1 by dt is equal to r1 n1 k1 minus n1 minus alpha 1 2 n1 by k1. dn2 by dt is equal to r2 n2 k2 minus n2 minus alpha 2 1 n1 by k2, where n1 is the number of individuals of species 1, n2 is the number of individuals of species 2, r1 the intrinsic rate of increase of species 1, r2 the intrinsic rate of increase of species 2, k1 the carrying capacity of species 1, k2 the carrying capacity of species 2, alpha 1 2 the competition coefficient effect of species 2 on species 1, alpha 2 1 effect of species 1 on species 2, and t the time. The outcome of competition depends on the relative values of k1, k2, alpha 1 2 and alpha 2 1. If k1 by alpha 1 2 is less than k2 and k2 by alpha 2 1 is less than k1, either of the species wins. If k1 by alpha 1 2 is less than k2 and k2 by alpha 2 1 is greater than k1, species 2 wins. If k1 by alpha 1 2 is greater than k2 and k2 by alpha 2 1 is less than k1, species 1 wins. And if k1 by alpha 1 2 is greater than k2 and k2 by alpha 2 1 is greater than k1, there is stable coexistence of species. The Lotka-Volterra competition equation has a drawback that competition coefficient is independent of density of interacting species, while studies reveal that competition coefficient is a function of density. There are 2 main effects of competition: competitive exclusion and character displacement on the basis of their short-term and long-term effect. The mathematical examination of phenomenon of competition by Lotka-Volterra led to the formulation of idea that 2 species with same or similar ecologies cannot coexist in same environment by Gauss in 1934, known as competitive exclusion principle, also known as Gauss's competitive law or 4C law, which implies completely competing species cannot coexist. Gauss studied the growth patterns of two species of Paramecium having Paramecium aurelia and Paramecium caudatum when cultured together and separately. Each species had a sigmoidal growth pattern when grown separately. However, when both species were grown together, their growth patterns were sigmoid in the first week, but later there was a gradual increase of Paramecium aurelia and gradual decrease of Paramecium caudatum. This experiment confirmed the competitive exclusion principle. Thomas Park also demonstrated the competitive exclusion principle using the green beetles Tribolium confusum and Tribolium castanium under different environmental conditions. The two species showed characteristic growth pattern in separate cultures, but in mixed cultures, one species excluded the other and the outcome was not always constant. Further experiments established that the outcome of competition depends on number 1, initial population densities, number 2, environmental conditions, and number 3, genetic constitution of the strains of competing species. Gauss in 1934 in his other experiment was able to show that by manipulating these factors, it is possible to create a situation in which two potential competent species, Paramecium caudatum and Paramecium berseria, can coexist. The population curves of these two species in separate culture were markedly different, and Gauss concluded that they could coexist because their ecologies were not sufficiently different enough to result in exclusion. Many other experiments have demonstrated sustained coexistence of species under certain conditions, while under other conditions there was exclusion. W. L. Brown and E. O. Wilson in 1956 came up with the theory of ecological character displacement. Character displacement is a phenomenon in which two species that live together in the same environment have evolutionary effects on each other and tend to diverge in those characteristics that overlap. Brown and Wilson in 1929 compared the characters of a number of species living together with characters in the same species living apart, or in allopatry. They found that sympatric species possessed many different characters, although these same species were sometimes indistinguishable when living allopatrically. Brown and Wilson concluded that these situations result from competition. As the species were similar, they competed for the same resources, and natural selection favored those species that competed less. Thus, the characters diverged. Darwin worked on finches of Galapagos Island. He observed that they had competition with one another, which led to adaptability to avoid competition. Their feeding structure changed with evolution, and thus their feeding habits became varied and restricted. Thus, it showed character displacement. Predation, herbivory, parasitism, represented as negative positive, are the biological associations in which one species benefits while the other is harmed. An organism which feeds on another organism for its food is called predator, while the organism that is fed upon is termed as a prey. This interaction between the prey and predator is known as predation. Russian biologist Gauss studied interaction of two ciliated protozoans, Paramecium caudatum and Dinidium nasutum. Paramecium thrives well on yeast and bacteria, while Dinidium thrives well on the diet of fresh Paramecium. In the first experiment, 5 Paramecium were introduced in a test tube containing an oat medium which is conducive to the bacterial growth. 2 days later, 3 Dinidium populations were introduced. 3 and 4 days later, respectively, both Paramecium and Dinidium populations were extinct. Variation in the introduction of predator and the variation in the size of the container produced similar results. He then modified the homogeneous oat medium by adding certain sand particles which created an effect called by Gauss's refuges where Paramecium caudatum could hide. The simultaneous introduction of predator and prey under such conditions was followed by continuous increase in the population size of prey and extinction of predator. From this study, Gauss concluded that as the ecosystem approaches the natural heterogeneous conditions, population control depends on multiple processes. However, The extinction of prey-predator relationships in a homogeneous environment can be mitigated to an oscillatory pattern by repeated immigrations. Lotka in 1925 and Volterra in 1926 independently derived a set of equations to describe the interactions between the populations of predator and the prey. We know that prey population can increase exponentially as the rate of change of population growth is equal to the multiple of intrinsic growth rate of prey R1 and prey population size n. dn by dt is equal to R1n. If R1 is positive, there is geometrical increase in the size of prey population. Without prey, the predator population dies off based according to the equation dp by dt is equal to negative R2p, where R2 is intrinsic growth rate of predator and p is predator population size. If the prey and predator are grown together, the rate of increase of prey is slowed by a factor depending upon the density of predator population. Rate of change in the population of prey is given by the equation dnh by dt is equal to RH NH minus P NH NP, where RH is host per capita increase, NH is exponential increase of prey population, NP is number of hosts killed by predator and P is predation rate. Likewise, the predator population will increase at the rate that depends upon the density of the prey species. Rate of change in the population of predator is given by the equation dnp by dt is equal to Cp NH NP minus Tp NP, where Cp is rate at which prey are converted into predator offsprings and dp is predator death rate. Thus, predator-prey have reciprocal oscillations. Herbivory involves the predation of plants or algae. The best documented case of plant herbivore dynamics came from introduction of animals used as biological control agents, where animals feed on plants such as weeds. One such example is the elimination of prickly Opuntia vulgaris due to the introduction of the insect Dactylopius salonix. The biological weed control systems also provide examples of cases where a herbivore regulates the abundance of a plant and is itself food limited. An example concerns Cardus natans, which infests thousands of acres of grazing land in the United States and Canada. A weevil, Rhinocylus conicus, which feeds on the seed heads of the thistle, was imported from France in 1969. After 3 years of introduction of weevil, the abundance of thistle reduced and the population of plant was maintained at equilibrium levels. Parasitism is an interaction when one organism feeds on another but does not normally kill it outright. The organism is termed as parasite and the prey is called a host. Parasites obtain their nutrients from one or more individuals, causing harm but not immediate death. Thus, parasite draws benefit from host, while as the host suffers decrease in fitness through decrease in survival and reproduction or growth. Host can be definitive host, in which parasite exhibit sexual reproduction, or intermediate host, which contains non-reproducing forms of the parasite. Insects are often host to other species of parasitic insects termed parasitoids. Parasitoids develop as internal parasites of insect immature states such as larvae or pupae of hosts. Some parasitoids themselves serve as host for other parasitoids, which are known as hyperparasitoids. Anderson and May have given a useful distinction of parasites into microparasites and macroparasites. Microparasites are microscopic parasites that multiply directly within their hosts, usually inside the host cells, for example, pathogenic bacteria and virus, while as macroparasites such as schizostomes are the parasites that are visible to the naked eye and live in the host but release infective juvenile states outside the host's body. Parasites can either be ectoparasites which live on the outside of the host's body such as ticks and fleas, or endoparasites which live inside the host's body such as pathogenic bacteria and tapeworms. Parasitic flowering plants may be classified as holoparasites or hemiparasites. Holoparasites lack chlorophyll and are totally dependent on the host plant for their water and nutrients. One famous holoparasite is the tropical Rafflesia arnoldi, which lives most of its life within the body of its host. One famous holoparasite is tropical Rafflesia noldi, which lives most of its life within the body of its host. Hemiparasites generally photosynthesize, but they lack a root system to draw water and thus depend on their hosts for their function. Mistletoe viscum album is a hemiparasite. Studies of Alexander and Burden have shown that rust caused by Albugo candida and downy mildew caused by Peronospora parasitica have significant effect on the survival and reproduction output of affected individuals of Capsula bursa pastoris. The systematic infection of seedlings from the seed-borne or soil-borne spores result in 90% death of plant, while as secondary infections during the later stages of development had a little effect on mortality. This example shows age-specific mortality due to parasitism. The use of host-specific pathogen in weed control provides some of the best evidences of impact of pathogen on plants. Best documented case is the introduction of rust fungus Puccinia chondrulina in Southeast Australia in 1971 to control skeleton weed Chondrilla juncea. This skeleton weed is a native of Mediterranean region with numerous forms having maximum density of less than 10 plants per meter square, while in Southeast Australia density can be even 1000 plants per meter square. Only 3 forms A, B and C grow there, of which type A was most widespread. In native range, weed was controlled by rust Puccinia chondrulina, which was then introduced in Australia where the rust had no effect on forms B and C, but it reduced the density of A form from 1000 to 10 individuals per meter square, which was followed by increase in B and C form. Thereby, competition of plants of type A previously restricted the abundance of B and C prior to the introduction of fungus. Commensalism, represented as positive neutral, is an interaction in which one partner benefits while the other experiences no benefit or harm. In this association, usually, a commensal can obtain nutrients from the host species for their shelter, growth and locomotion, and the host remains unaffected. The host is larger and unmodified, while the commensal is small with some modified structural adaptations with its habitats. Indirect commensalism occurs when the activities of one species indirectly through an intermediary species benefit another species without itself being helped or harmed. Gregory Martinson, Elizabeth Dreeb and Thomas Witham of North Arizona University studied an indirect commensalism in which beavers Castor canadensis indirectly benefit a herbivorous beetle species Chrysomela confluens through their effects on cottonwood trees popular species. Searching for a mechanism to explain the higher beetle concentration in areas where beaver foraging activity was greatest, they found that compared to leaves on undamaged cottonwood trees, the leaves on stump sprouts had higher concentration of the defensive chemicals used by cottonwood trees to repel beavers and other herbivorous mammals. However, beetle is not repelled by the tree's defensive chemicals and instead uses them for its own defense. In addition, the leaves on stump sprouts were also higher in nitrogen compared to the leaves on uncut trees, making them a better source for the beetles. Some examples of commensalism are lianas, epiphytes, epizoans, epizoite, etc. Lianas are vascular, woody plants rooted in the ground which climb up with the support of other trees and reach almost at the top of the forest canopy. The woody stem of lianas is closely attached to the supporting tree, but is not involved with it in any nutritional relationship. Bauhinia wahili is a common liana. Other common lianas are species of ficus and Ctenospora. Plants that grow on other plants only for support and not for water or food supply are known as epiphytes. They differ from the lianas in that they are not rooted into the soil. Certain common examples of epiphytes are orchids, bromeliads, hanging mosses, usnea and electoria. For meeting water requirements in the epiphyte distichia, the leaves fold to form a jug-like structure having a narrow mouth where water accumulates. Roots of the epiphyte enter and grow in these specialized cavities of leaf. Epizoans are plants that grow on the surfaces of animals. For example, some green algae grow on the long grooved hair of sloth. Basiclidia grows on the back of freshwater turtle. Likewise, epizooid animals are associated with another animal for the purpose of anchorage and protection, such as the protozoans Trichodina and Cherona on hydra, water-celids or various other aquatic organisms. The barnacles, which attach themselves to the back of the whales, and shells of horseshoe crabs and so on. Commensals may also be internal. For example, many harmless protozoans occur in the intestinal tract of mammals, including man. Some microorganisms such as bacterium Escherichia coli is found in the human colon. The hermit crab normally lives inside gastropod shell. The pitcher of the pitcher plant found in bogs furnishes a breeding site or home for certain species of midge, flies, mosquitoes and tree toads. Mutualism, represented as positive-positive, is an interaction in which both partners reciprocally benefit from the presence of each other. Mutualism can be defined by many factors. Depending on the degree of association between the species, the mutualism may be symbiotic or asymbiotic. Depending on the degree to which the interaction is necessary for the survival of each species, it may be an obligatory or facultative. This relationship may be further defined on the type of benefits that are drawn from such an association, for example, protection, dispersal, etc. There are 4 types of mutualism. Number 1, persistent type. Mutualism where mutualists have very tight symbiosis. For example, rhizobium with legume. Number 2, intermittent type. Mutualism in which mutualists do not have a tight relation but interact intermittently for the limited period of time with interactions reoccurring at predictable intervals. For example, pollination of flowers by bees. Number 3, sequential type. Mutualism in which one or both mutualists have association, leave and are able to establish new associations with another species and so on. Number 4, diffuse type. Mutualism in which any number of species singly or simultaneously interact with each other. Some examples of mutualism are pollination by animals, lichens, etc. Pedicularis and Dactylorhiza have same color and shape flowers to deceive the bumblebee. Lichen is made up of a fungus within the cells of which an algae is embedded. The fungus absorbs the moisture and nutrients while as the algae manufactures the food. It is an obligatory type of mutualism. Mycorrhizal association between fungus and roots of higher plants is also a mutualistic association. Mycorrhizae are of 2 types, differentiated by the fact that the hyphae of ectomycorrhizal fungi do not penetrate individual cells within the root. For example, species belonging to Pinacea, Betulaceae, Salicaceae and Myrtaceae, while the hyphae of endomycorrhizal fungi penetrate the cell wall and invaginate the cell, found in Ericaceae, orchidaceous plants and vesicular arbuscular mycorrhiza. Amensalism, represented as negative neutral, is an interaction in which one species is harmed while the other one is neither benefited nor harmed. Amensalism, which commonly involves some type of chemical interaction by which the organisms of one species affects the well-being and growth of individuals, is called allelochemic. The inhibiting substance may be inorganic chemicals such as acids or bases, organic toxins or antibiotics. The majority of inhibiting chemicals are produced as secondary substances by plants and released into the soil through the root or leaf wash. The suppression of growth through the release of chemicals by a higher plant is known as allelopathy. Thus, allelopathy, allelon, which means each other, and pathy, which means suffering, means chemical control of distribution among plants. For example, walnut tree produces a non-toxic substance, juglone, which is found in its leaves, fruits and other tissues. When the leaves of fruits fall on the ground, juglone is released to soil, where it is oxidized to a substance that inhibits the growth of certain understory species and garden plants such as heaths and broadleaf herbs and favors other species such as bluegrass and blackberries. Penicillium, a fungus found in soil, produces antibiotic substance that check the growth of a large variety of bacteria. A number of antibiotics such as penicillin have been used extensively in human medicine. Antagonistic substances are also reported in some algae. as for example in culture of Chlorella vulgaris, some substance accumulates which inhibits the growth of diatom Nischia frustulum. Pond blooms of blue-green algae, especially microcystis, are known to produce toxins such as hydroxylamine, which causes death of fish and cattle. With that, we come to the end of this lecture. I hope you understood. Thank you and goodbye.